Hi, everyone, and welcome to today's lecture on enzyme mechanisms. Now, the first thing I want to point out when we talk about enzymes is depending on which class you are in or which field you are in, you might hear slightly different terminology. So, when you hear catalysis or catalytic, that basically tells you the process of increasing the rate of chemical reactions. So, you will hear of catalysts if you are in, for instance, a chemistry class. Whereas in biological fields, we say enzymes, which are biological catalysts. And they're usually going to be globular proteins. So whenever you hear enzymes, I want you to think of proteins, which is important later on when we talk about things like temperature, because as you know, proteins can become denatured. But I want you to put little stars here next to the exception. So enzymes are usually proteins with the exception of self-splicing RNA. Okay, so make sure you know that the exception is self-splicing RNA. Now, when we talk about enzymes, the critical aspect of enzymes, in order to work, they have to be able to bind a substrate. And so over here, when I have the question marks, I want you to fill in that a small part of the enzyme called the active site, so here it is over here, the active site, will bind with these substrates. And the substrates, which you see here, adorable in this little image here, the substrates are the reactants. Now, the enzyme is going to be critical to bringing those guys close together so that they can interact and react. Now, when we talk about the substrates binding to the enzyme, there are two different models that I want you to be aware of. In the lock and key model, which is shown over here, and this one's supposed to be moving too. Oh, it is moving. Okay. Sometimes that works, sometimes it doesn't. So in the lock and key model over here, that shows the shape of the enzyme's active site is very specific to the shape of the substrate, and it's not going to change, and it will only bind the substrate that is specifically matched to that shape. The induced fit model, on the other hand, shows that the active site, it's very tough to see, but it's see how it's changing a little bit? The active site will sometimes undergo a slight change in shape to better fit the substrate. Okay. And the way we look at these models, it's not that, one existed and then we replaced it with the other model. It's that some enzymes will follow the lock and key model and other systems will demonstrate the induced fit model, which tends to be more the currently accepted model now. Now, when we talk about enzyme action, I want you to circle star highlight the fact that enzymes speed up reactions by lowering activation energy. So over here, this question mark is lowering activation energy. And the activation energy is the peak that you see in these graphs here. Okay, the activation energy is basically the amount of energy needed to start the reaction. And what you notice here is in red, you have a very high activation energy required without an enzyme. Then when you add the enzyme, it's much lower. Now, the way that I like students to visualize that is basically think of if you want to buy a cell phone. Let's say there's a really great new cell phone that you want and it's $1,000. And so you'll have to save up for a while before you can get that cell phone. Let's say, however, all of a sudden there's a big sale and it's only going to cost $100 right now. Well, now you can suddenly get it. You don't have to save up anymore because now they made it more attainable by lowering the starting price for you to get it. So that's what enzymes do. They lower the starting energy needed so that you can have the reaction start sooner. It speeds up the reaction. Now, notice that in doing this, what we call the free energy, the overall free energy, which is the energy difference between reactants and products, so that's the difference over here, that does not change. Okay, so enzymes lower the activation energy, which is seen over here, but do not change the overall free energy, which is the energy difference between the reactants here and the products here. Notice that the green and the pink line are the same distance over here, whereas here it dropped down when you have the enzyme. Now, when we talk about how enzymes are working, I just want to give you another quick example to have in your mind about how they actually work. So, the reason why enzymes are able to speed up a reaction and help a reaction out is I'd like you to picture them as if the enzyme is a seat on a school bus. And so when you think about this, picture how, okay, you have these two little kids getting on the bus. They don't know each other. It's the first day of school. When they sit next to each other on that school bus seat, because they're right next to each other and in close proximity to each other, what's going to happen? Well, they'll start to chit chat. They will start to interact and react with each other. And so by the time that they get off of that school bus, they're like, oh my God, we're best friends now, besties for life. And so when you think about that, those two little kids that have now bonded by being in close proximity to each other, they leave that school bus, and what do you notice? That seat is the same as when they came in, okay? Or at least I hope it is. And so the seat is not used up in that interaction, that reaction. And instead, now the next set of students can come onto that school bus and sit in that seat, be close together, and interact and react. And so the seat does not get used up. And so that's what enzymes do. They put two substrates into close proximity or, close to each other, and so that those substrates have a nice place to interact and react. Because think about it, if someone's in a different classroom than you, you're less likely to interact to them, right? You're not going to start shouting down a hallway to talk to people in the next classroom. So enzymes put things close together so that they can react and interact and speed up that reaction and that bonding. Now, in order to do their job, enzymes need a little help. They need to be activated. And so I want you to know all of these terms and be able to distinguish the differences between them. So, for instance, the difference between cofactors and enzymes, or the difference between those two and prosthetic groups, or the difference between apoprotein and holoprotein. So, the first one is cofactors. And when you hear cofactors, I want you to visualize a steel factory. And the reason why I say that is when you see factor, think steel factory. Cofactors are inorganic molecules, including metals and minerals, that are needed to help supplement and activate an enzyme. This is why it's so important to have supplements or in your diet things like zinc and magnesium. Okay, very important cofactors for your enzymes and your metabolism. Coenzymes, on the other hand, these do the same thing. They're activating enzymes and they're free floating, just like cofactors, but these guys are organic. So, coenzymes, I want you to write down organic molecules, including vitamins. Okay, now both of these cofactors and coenzymes, both of these are free floating molecules. So they're separate from the enzyme, but they will activate and help the enzyme work. That's different from prosthetic groups because prosthetic groups are permanently, sometimes, covalently bonded to the enzyme. So these can be either organic or inorganic, and they are bound tightly to the enzyme. Okay, and that's how they're different from the first two terms. Then we have apoprotein and holoprotein. And I apologize in advance because the way that I help students remember these is by using the highly offensive, stereotypical accent of my ancestors, the Italians. And so my Italian family members who would kill me if they heard me teach this lesson. So apoprotein, when you have a in front of words, it means, without. So apoprotein is when you have the enzyme without any of the cofactors or coenzymes or prosthetic groups. And since it's just the enzyme without any of these helpers, it is inactive. Okay, apoprotein is the inactive enzyme. Then holoprotein, the way that I have my students remember it, again, I'm so sorry for this, is think holoprotein. It's the whole thing, the whole enzyme plus all of the cofactors or coenzymes and prosthetic groups needed to activate that enzyme. And so the holoprotein is active, the apoprotein is inactive. One of the coenzymes that comes up the most in this particular class in biochemistry and in your metabolism for all of the different metabolic reactions is called NAD plus and NADH. Okay, so you see it has two different forms: NAD plus when it's oxidized, NADH when it's reduced. And these guys are critical. Okay, their official name is nicotinamide adenine dinucleotide, but I'm not going to make you memorize that. What I want you to know though is that NAD is the active form of vitamin B3, also known as niacin. So that's why vitamin B is so important for you. That's why niacin is so important for you. But don't overdo it. Anything in excess can be toxic for your body. But that's why it's so important, because NAD and NADH, these function in helping out or being coenzymes, activating the various redox reactions that we're going to talk about when we go through each form of metabolic processes in this course. And so when you look at them, I want you to know that NADH functions as an electron carrier that will donate a pair of electrons to redox reactions, whereas NAD plus is an electron carrier that will accept a pair of electrons and oxidizes substrates. Okay, now the trick to remembering which is when you look at them, notice NADH versus NAD. This one has an extra letter. So you can think of it as, oh, this one has extra, so it wants to give away. So NADH will donate electrons in reactions, whereas NAD plus will accept electrons in reactions. And they're also uniquely called co-substrates sometimes because, like I mentioned before, a lot of times when you're dealing with enzymes or even coenzymes and cofactors, they're usually not changed in reactions, but as you see on this slide, these two are slightly altered. It's going from NADH to NAD plus. So they are called co-substrates in addition to being called coenzymes. Now, most people know that when you increase temperature, you increase the rate of reactions in chemistry. Because think about it, the hotter something is, the more you're going to have molecules and atoms moving. They're going to speed up, they're going to bounce into each other more. The more they bounce into each other, the more reactions you have. But when we talk about enzymatically controlled reactions, okay, so this second part here, you notice what happens to the curve is once you reach a certain temperature, the amount of activity plummets. You could even completely stall or stop the reaction. So you can make the reaction go faster as you increase temperature, but when it's enzymatically controlled, you hit a point where now a higher temperature will stall or stop the reaction. And that's because we mentioned earlier enzymes are proteins. And what happens to proteins at higher temperatures? They get denatured, which means they unfold and now they are not going to function anymore. Now, some of the ways that enzymes increase the rate of reactions, I basically gave you the general overview of how to think of it, but the more specific case is that they're basically stabilizing what's called the transition state. And transition state is when you have an enzyme substrate complex and you have substrates interacting. And so they they can be considered creating an alternate path for product formation and reducing entropy by orienting the substrates appropriately. But overall, the key here that I want you to remember is over here, why is the active site significant to this? Well, this active site, which like I said is like that seat that the students are sitting on, that active site is providing the optimal physical and chemical environment for the substrates to interact, for the products to form. Okay. And so it's giving a nice, safe environment for you to have a reaction occur. So, a moment ago, I mentioned what's called the transition state, and that's when you're transitioning from the substrates or reactants into the products. And so you have that middle area where you have the enzyme substrate complex and the substrates are currently interacting. Now, what's important about the transition state is that the enzyme will have a higher affinity for the transition state in the middle here compared to either the initial reactant or substrates and the products. And what that means is when an enzyme, when something has high affinity for something, it means it wants to be bound to it. It absolutely adores it. It wants to be stuck to it. It wants to follow it around. Basically, like the students that I can't get rid of and they just want to follow me around all day. Yeah, so that's high affinity for something. So because the enzyme has a high affinity for this transition state compared to the products, that means that it will hold on tightly to the substrates when the reaction is going on. And then when the reaction is finished, now it has a lower affinity, which means it lets go. So it releases the products. So that's the impact or the significance is that the higher affinity for that transition state compared to the products allows for the products to then be released at the end. Now, I know these next few slides will look absolutely terrifying, totally scary, but I am not going to make you memorize the details of these reactions or mechanisms. I just want you to be aware of the various catalytic mechanisms that exist and the general idea. The first one is acid-base catalysis. And with this concept, when you think of acids and bases and you think of pH, you think of hydrogen, right? Protons. And so, with that, acid base catalytic mechanisms is basically going to include a proton transfer. It'll either involve water or a functional group, but the main idea is that you will see the transfer of a proton occurring. And so, in this case, you see water and you see the hydrogen representing the proton highlighted in pink in this case. Whereas the first mechanism we just mentioned dealt with a proton transfer, this one here, covalent catalytic mechanisms, deals with the transfer of electrons. And so basically, what you have is a nucleophile group and an electrophile group. So, a nucleophile will donate electrons as an electron pair, and the electrophile, think of it as electrophile, phile means you like something. So electrophiles like electrons, so they will take the electron pair, and we call it a nucleophilic attack a lot of times. So basically, you'll see the term nucleophilic attack, or you'll see the transfer of electron pairs using our good old buddy, the coenzyme NADH NADH, and then you also see NAD plus present as well. And so whenever you see that, we're referring to covalent catalytic mechanisms. Then we have the metal ion catalytic mechanisms, and this one's, most straightforward in the title of it. Metals are basically used to promote the orientation, putting the bound substrates in proper orientation and helping in the redox reactions. In the scenario in the figure here, you see that the metal used here is zinc, but it can be any of the metal ions basically. And so, just to recap the ones that we mentioned, here's an example of taking a look at enzyme mechanisms and how you could have more than one occurring at an active site as well. So, this is taking a look at chymotrypsin, which we hear a few times in this course. And so, when you look at the figure here, you'll notice a couple of things. First of all, the moment you see nucleophilic attack, I want you to circle that in your notes. Nucleophilic attack tells you that we are dealing with a transfer of electrons, which would be covalent mechanism. And then you also see the donation of a proton. So that tells you acid base catalytic mechanism. Okay. So I want you to circle nucleophilic attack because that tells you that chymotrypsin is using the covalent mechanism, as well as circling the donation of a proton, which tells you acid base catalytic mechanism. Now, the many, 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 many different enzyme mediated reactions can basically be grouped into three main categories. We have coenzyme dependent redox reactions, we have metabolite transformation reactions, and we have reversible covalent modifications reactions. And you're going to see all of these in the different chapters that we look at where we focus on individual metabolic pathways. So the first one, coenzyme mediated. Well, when we looked at the main coenzyme of NADH and NAD+, we talked about electron carriers. So in coenzyme dependent redox reactions, you'll have the requirement of coenzymes functioning as electron carriers. The metabolic or metabolite transformation reactions. Those will be for anabolic and catabolic pathways involving terms like isomerizations, condensations, and dehydration or hydrolysis. And you're going to see that when we talk about steps of pathways like glycolysis, for instance. Then the reversible covalent modifications. Reversible tells you that it's kind of like having a molecular switch turning off and on cell signaling and gene expression. The main one that I want you to remember for this one is the use of kinases and their opposite, which are phosphatases. And so kinases will add a phosphoryl group, phosphatases will remove it. And a lot of times that phosphoryl group is coming from ATP. So now we're going to take a look at enzyme kinetics. We're not going to go into crazy detail here, but I want to point out a few things. Uh, the first thing that I have here that I ask you is how do we demonstrate the conversion of a single substrate to a single product? And that's this equation here. Okay, so the answer to this first question is this equation here, S being substrate, P being product. Then for an enzyme catalyzed reaction, we have this big formula here. Again, I'm not going to make you memorize these formulas. I just want you to be able to recognize what they mean when you see them. So this tells you enzyme plus a substrate then forms the enzyme substrate complex, which is the transition state. And then you have the release of the products and the enzyme being separate now. I want to point out that you'll notice that K to the subscript 1 is the rate constant for the formation of the enzyme substrate complex, so you see it going in this direction. Whereas the other two values you see, K subscript 2 and K subscript -1, notice that they're going away from the enzyme substrate complex. So that means the dissociation of the enzyme substrate complex, or breaking apart the enzyme substrate complex. Okay? So make sure you remember what each of those K1, K-1, K2, what they represent. Now, when we look at Michaelis-Menten approach on a graph, I want to point out a few things. One of the things that we're going to take a look at is K subscript M, which is the Michaelis constant. That basically is a numerical value to represent the strength of binding a substrate to an enzyme. And we're going to see in the next figure how we can actually take a look at that. And don't worry, I don't want you to memorize this formula. I just want you to kind of know some of the terminology that I'm going to go through in the next few slides. So over here, I actually had it in the chalkboard notes a moment ago, but over here, I just want to point out that when we're talking about the Km, or the Michaelis constant, you notice that it is when the rate of the reaction is half of its maximum value. This, we take a look at the substrate concentration, okay, and that gives us the Km. So if we have here the overall maximum value of the rate of the reaction, when you go to half of that, you then find the substrate concentration, and that gives you the Km here. Okay, on this slide, two things I want to point out. You notice that the curve flat lines. The first portion, the increasing portion, is called first order kinetics. That is substrate concentration dependent. Then it flatlines, okay, flatlines over here in zero order kinetics, where now the rate is independent of the concentration of the substrate. The reason that is that at some point you have now filled up all of the active sites of the enzymes present. So you could have a ton of extra substrate, but it's not going to increase the reaction rate, or the velocity of the reaction, because those substrates have nowhere to bind. You have saturated all of the enzymes. Okay, so that's why the rate flatlines in zero order kinetics. The rate no longer depends on concentration of substrate because all of the enzyme active sites are full. Okay, so the reason why I emphasized knowing K1, K-1, and K2, what they represented, is to make this portion a little bit clearer to you. So again, remember K1 was the rate constant for the formation of the enzyme substrate complex, whereas K-1 and K2 were both representative of the different ways that you can split up or break down that enzyme substrate complex. So now they represent it over here. So when you see negative, that's breaking it down. When you see positive, that's building it up. And the steady state is this equation here, which tells you that the rate of formation of the enzyme substrate complex is equal to the rate of breakdown of the enzyme substrate complex. Now, the next concept that I want you to be aware of are the Lineweaver-Burke method graphs in order to calculate Km and Vmax. And so with these double reciprocal plots that they call them, you basically have a way to analyze the kinetics of enzyme catalyzed reactions. So when we look at this graph, I want to emphasize here and here, the reciprocal of the y-intercept. So meaning if you do 1 over y intercept, so 1 over whatever number your line crosses on the Line-Weaver-Burke method over here, that would give you the Vmax, okay? Because it's basically you're turning this equation into solving for Vmax, which gives you 1 over the y-intercept is Vmax. Then -1 over the x-intercept gives you Km, the Michaelis constant. So to demonstrate that idea with actual numbers, here we have a Line-Weaver graph. And so we said that the reciprocal of the y-intercept, meaning 1 over y, would give us Vmax. So if you look at this graph, the y-intercept is about 12. So 1 over 12 would be our Vmax. Then you notice that the x-intercept is approximately minus 0.15 or minus 0.155. Okay, so the Km would be -1 or -1 over negative 0.15 or 0.155. Okay, whatever the intercept is over here. So now when we talk about enzyme mechanisms and mechanics, it's important to think about enzyme inhibition because, a lot of times you don't want enzymes just working nonstop. You need a way to control them. So there's four main types of reversible inhibition. So keep in mind, when you see any of the terms that we mention on this slide, such as competitive, um, pure non-competitive, mixed, or uncompetitive, any of those, it's going to be reversible. So the first one that we have is competitive inhibition. That's basically when you have a decrease in enzyme activity because there's the binding of a competitive inhibitor in the active site. So it's physically blocking the substrate from being able to access the active site. Okay, that's competitive inhibition. For non-competitive inhibition, there are two forms that I want you to be aware of. Okay, there is the pure non-competitive. Okay, and basically in non-competitive, you notice that the inhibitor is binding somewhere that is not the active site. The active site is over here. The inhibitor is binding elsewhere and causing an allosteric change, meaning a change of shape. So notice that now the active site shape has changed, so the substrate doesn't fit there anymore. Now, in pure non-competitive inhibition, the binding of the inhibitor has no effect on the affinity for the substrate. So it doesn't affect how well that enzyme or that substrate wants to bind the enzyme. What basically happens is that the inhibitor, when it's bound, c- catalytic effects cannot occur. And so that's basically very rare because what's more common is called mixed. So you see that over here, mixed non-competitive inhibition. That's when the inhibitor does affect the binding of the substrate. And, it affects the enzyme's affinity for the substrate, which is much more common. Pure non competitive is very rare, um, and mixed is a lot more common. The last one is uncompetitive, and you notice that the inhibitor can only bind to the enzyme substrate complex. So it can only bind when the substrate has already bound to the enzyme. It can't bind to a free enzyme. So you notice over here and here, the inhibitors are already bound to just the enzyme without the substrate. So that's the difference with that one. Now, when we take a look at Line-Weaver-Burke plots, you're able to tell which form of inhibition is occurring. And so the first one over here, this one is competitive inhibition. These are competitive inhibitors. You notice that When you have no inhibitor and then when you add inhibitors, the lines are crossing on the y-axis. So if you see the lines intersecting or crossing each other on the y-axis, that tells you it's competitive. The next one over here, you notice that they're not crossing, they're not touching on the Y. These two lines, the without inhibitor and with inhibitor lines, they meet up at the x-axis is where the two lines intersect. When you see them meet up at the x-axis, that is pure non-competitive. So for this one, write pure non-competitive. This one is mixed non-competitive. It's where they are intersecting on neither the x nor the y. Instead, they are intersecting in the left quadrant. So they're intersecting over here, not on the axis. The last one is uncompetitive. So this one here is uncompetitive. It's when you see two parallel lines, the without inhibitor and the inhibitor lines are parallel. They never intersect. They never touch each other. Now, all four of those, the competitive, the pure non-competitive, mixed non-competitive, and uncompetitive, were all reversible. Now I want to mention irreversible inhibition, and the examples that this would include is the antibiotic penicillin, as well as cyanide gas, heavy metals, and nerve gas. So in the figure here, you have penicillin, and you also have heavy metals such as mercury being able to irreversibly inhibit enzymes. And the way they do that is through covalently binding. So the inhibitor will covalently bind to the enzyme, causing permanent inactivation. So you notice here, the heavy metal is now permanently bound to the enzyme, inhibiting it. And over here as well, it's now permanently bound, covalently bound to the enzyme. And so in the case of penicillin, circle star highlight that that is a case of irreversible inhibition. Basically, penicillin will bind to the enzyme of bacteria that the bacteria needs to build their cell wall, to build the peptidoglycan of their cell wall. If you bind to that enzyme, you now kill the bacteria. And that's a great way to be selectively toxic when you're giving a drug because you're targeting something that the bacteria have that we do not have because our cells do not have a cell wall. Now, I want to mention, in addition to the Michaelis-Menten form of enzymes, we also have allosteric enzymes. That basically means enzymes that have an additional site in addition to their active site. And so, for instance, if an enzyme has an inducer or a repressor site, kind of like you see in genetics or in your intro bio classes when you talk about the lac operon and the trip operon in bacteria. When you have a site that's separate, so this site here is an allosteric site separate from the active site of the enzyme. And over here, you have the two sites as well. They can either activate or repress the enzyme or a pathway and that is allosteric. It triggers a change in shape of the enzyme. And so these guys are controlled by feedback inhibition or also inhibitors can block them. And what we mean by feedback inhibition is when you have a pathway. So most things in the body, most metabolic reactions occur in pathways. And so the final product can end up being an inhibitor to the initial enzymes. And so that tends to happen with allosteric enzymes. Now, I just want to point out that you can't think of inhibition of these allosteric enzymes in the same way that we talked about the other ones. I'm not going to go into crazy details with these, but I just wanted you to be aware that you won't see terms like competitive or non-competitive inhibition. You won't see the Michaelis constant, so the Km. You see different descriptions when it's allosteric, okay? But we're not going to go into details of allosteric enzymes. Now, earlier I mentioned molecular switches in terms of ways that enzymes can function or be controlled. And so I just want to point out kinases and phosphatases. So enzyme activity can be controlled by phosphorylation. You notice in these figures that kinases will add a phosphoryl group, whereas phosphatases are the opposite. They remove a phosphoryl group. But what's interesting is that phosphorylation across the board does not always increase or decrease enzyme activity. So it depends on the enzyme. You can't say that, oh, kinases will always activate enzymes or phosphatases will always deactivate. That's not true. It depends on the enzyme, and so we can't predict whether phosphorylation will increase or decrease the activity of an enzyme since it varies from enzyme to enzyme. Now, I just want to give you a few interesting terms before we finish off this lecture. The first one is zymogens, and I have a highly inappropriate way of remembering what these do. So, zymogens are basically proteins that are formed or synthesized as inactive and then that get activated by specific hydrolysis of their bonds. So, proteolysis. And another way of saying that is cleavage. So the way I like to tell students to remember zymogens is that zymogens are inactive proteins that are turned on by cleavage, just like a lot of people I know. And so when you think of zymogens, think of the two big examples of chymotrypsin and trypsin. Okay. And basically, when you think of examples like this, these are, um, they're formed in the pancreas and they are highly digestive. So they're proteolytic, um, or they have protease activity. So if you formed chymotrypsin and trypsin in the pancreas and they were active, they would end up doing major damage to the pancreas. Instead, you need them to become active only when they're in the small intestine, where their digestive properties are needed. And so that's where they then get activated by cleavage, okay, of specific bonds. So trypsinogen and chymotrypsinogen, which are the precursor molecules of trypsin and chymotrypsin, they're zymogens that will not be active in the pancreas, but then become active in the small intestine because we don't want them doing damage when they're in the wrong place. The similar concept is caspases. So caspases are another example of zymogens. These are cysteine proteases that are basically responsible for a lot of different things in the cells. They're involved with signaling, with the immune system and stem cell differentiation. But the one that I want you to really remember for caspase function is they are involved in programmed cell death, meaning apoptosis. And so, if you think about that, you want these proteins to be first produced in an inactive form called procaspases, because if they were active right when they were formed, they would trigger apoptosis. They would cause the cell to die. So you need them to only be activated under certain conditions. Then we get to the last thing I want to mention, which are abzymes. So we talked about zymogens. Now these are abzymes. You can see everything to do with enzymes. They throw zyme in. And we already talked about in the first lecture of biochem that Zyme comes from German for yeast because enzyme, they coined that term since it was in yeast. And so abzymes are very important. I want you to circle star highlight this slide. Abzymes are antibodies with catalytic activity, meaning they are both antibodies and enzymes. Now, this is very valuable because if you think about it, antibodies are very specific, okay? And enzymes are able to do a lot of important functions like breaking things down, right? So, if we use abzymes in the medical field, well, one clinical example is in the treatment of cocaine addiction. And we already mentioned in another lecture how cocaine, what that ends up doing is it inhibits the transporter proteins that are supposed to take dopamine off of the receptors, off of the synapses, and you would then, not have this uncontrolled high. Cocaine blocks those transporter proteins, and so it keeps dopamine stuck on the receptors at that synapse. So you are getting this uncontrolled high, and you can't get that dopamine, you can't get the dopamine off of the receptor. So with this kind of situation, you can't, let's say, give a drug to block the dopamine receptors because, well, they're already taken up. That's the problem, is that you can't get the dopamine off of them. So what abzymes have done is you make an abzyme that's specific to the dopamine, okay? And you're able to have sorry, to the cocaine, you're able to have this targeted breakdown of the cocaine, and the cocaine is currently blocking the recycling of dopamine. It's blocking the transporter proteins from being able to take away the dopamine. So if you do the abzymes targeting the cocaine, the antibody portion will target the cocaine, and the enzyme portion will break down the cocaine into two safe products. Now the transporter proteins are no longer inhibited and they can pull the dopamine off of the receptors. Okay, so again, just I really want to make sure you understand this concept because abzymes are very powerful in the medical field. So, cocaine blocks the transporter proteins that would pull dopamine off of their receptors. Okay, so you don't want them staying uncontrollably stuck to the receptors. So cocaine currently is blocking the recycling or pulling the dopamine off. So now the abzyme will target the cocaine specifically, break it down into two safe products, which are shown here, uh, benzoic acid and I always mispronounce that one, ecogonic methyl ester. And so now that the cocaine has been broken apart, the transporter proteins are no longer inhibited and they can do their job of removing the dopamine from the receptors. Okay, that was a lot of information, and that is it for today. Any questions, as always, never hesitate to ask me morning, noon, or night. I have no life. I don't mind answering your questions in the Remind app. And please always send a picture of the slide that you are referring to. It makes it a lot easier for me to make sure that I'm helping you with the proper answer. Thank you and have a great day.